Okay, so welcome to this next video in which we're discussing the acute inflammatory response and anti-inflammatory drugs. Okay, so we've now discussed the three main blood vessels of the microvasculature, these terminal arterioles which split into capillaries and then into postcapillary venules. We want to now discuss how these pro-inflammatory mediators, interleukin-1, tumor necrosis factor alpha and histamine, which have been released by the sentinel cells, are going to act on the endothelial cells of these three blood vessel types. Okay, so they act differently basically. Histamine is going to act very, very quickly. Okay, so histamine is going to act on the endothelial cells and it's going to cause something known as type 1 activation of endothelial cells. Okay, whereas interleukin-1 and tumor necrosis factor alpha, they're going to take much longer to act, and to emphasize how much longer, histamine will act in minutes. Type 1 activation occurs in minutes, whereas interleukin-1 and tumor necrosis factor alpha, they are going to go to the endothelial cells and they'll both arrive in the same amount of time. So it's not the fact that these are just taking longer to get there, it's that the actual pathways interleukin-1 and tumor necrosis factor alpha induce take longer to initiate. Okay, and they're going to cause what's known as type 2 activation of endothelial cells. And this takes hours. So this one takes minutes, this one takes hours. So histamine activating type 1 activation is going to occur first. So let's start by looking at type 1 activation, since that's the one that generally in the acute inflammatory response occurs first, even though to an extent type 2 activation is more powerful. Okay, so we'll look at type 2 activation second, because that's the chronological order that things are going to happen in. So let's see the effect of histamine on an endothelial cell then. So histamine is going to come to these endothelial cells, so it will diffuse from the mast cells which have released it to the endothelial cells of the blood vessels in the infected area, and it's going to act on histamine 1 receptors which are within the membrane of these endothelial cells. So here is an H1 receptor. So this is an H1 receptor, and it's a GQ coupled, G protein coupled receptor. So H1 is short for the histamine type 1 receptor, or just the histamine receptor 1. Okay, so it's a G protein coupled receptor, so it has this typical 7 transmembrane structure where it has 7 membrane spanning alpha helices. Okay, and it's going to be coupled to a GQ heterotrimeric G protein. So let's draw the heterotrimeric G protein here. So heterotrimeric G proteins are named after which alpha subunit they have. However, they do have 2 other subunits. So they're made up of these 3 separate subunits here. An alpha subunit, a beta subunit, and a gamma subunit. And when you're building a heterotrimeric G protein, you have a huge scope for making many different heterotrimeric G proteins. So when you choose an alpha subunit, there are actually 16 different alpha subunits that you have available to choose from. When choosing a beta subunit, you have 5 different beta subunits you're available to choose from. And gamma, you have 12 different gamma subunits that are available for you to choose from. So there is a huge amount of scope for making different heterotrimeric G proteins. Now, if the whole G protein is called a GQ heterotrimeric G protein, then all that tells you is that the alpha subunit is alpha Q. So it tells you that of these 16 alpha subunits that you had available to you, you specifically chose the alpha Q subunit for your alpha subunit. Okay, it does not tell you what your beta and your gamma subunit are. Right, so as with all G proteins, heterotrimeric G proteins have an off state and they also have an on state. Okay, now at first this GQ heterotrimeric G protein is in the off state, and that means that the alpha Q subunit has GDP bound to it and is still bound to the beta and the gamma subunits. However, when histamine comes in and binds to the H1 receptor here, so this is histamine coming in from our mast cells, it's going to bind to the H1 receptor, and the H1 receptor will then become active, and what it's going to do is it's going to chop off this guanosine diphosphate from this alpha Q subunit. It's going to grab a guanosine triphosphate from the cytoplasm of the cell. So here is just a wandering guanosine triphosphate. It's going to stick that guanosine triphosphate onto the alpha Q subunit. Okay, so here now is our alpha Q subunit, which now has a GTP molecule bound to it instead of a GDP molecule bound to it. And when it's got GTP bound to it rather than GDP, then the G protein goes into the on state. And in the on state, the alpha Q subunit no longer wants to associate with the beta and the gamma subunit, so it cleaves away from this beta and this gamma subunit. However, they remain bound together and are henceforth referred to as the beta-gamma subunit. Okay, so we have this alpha Q GTP as it's called, so this is often referred to as alpha Q GTP, and we have this beta-gamma subunit that have been produced by histamine binding to the H1 receptor. So what we want to now see is, what do both of these do? So usually when you study G protein coupled downstream pathways, you focus completely on this alpha subunit and they abandon the beta gamma subunit. It's not interesting compared to the alpha subunit. Well, this is going to be one response where beta gamma actually does play a role. However, as always, we'll start off with the alpha Q GTP and we'll come back to beta gamma later. Okay, so what is alpha Q GTP going to do? Well, it's going to do what it always does. It's going to go and activate phospholipase C, specifically phospholipase C of the beta type. Okay, so in the phospholipid bilayer of the endothelial cell, you have an enzyme known as phospholipase C, and it's specifically phospholipase C of the beta type. So here is this enzyme which is sitting within the phospholipid bilayer. Okay, so this is phospholipase C, and it's specifically a phospholipase C of the beta type. So there are many different phospholipase C enzymes. This is phospholipase C beta that we are specifically activating from this alpha Q GTP subunit. So this alpha Q GTP subunit is going to go and it's going to bind to this phospholipase C beta and it's going to activate it. So let's colour a few of these in to make it look more interesting. So we'll colour phospholipase C beta in red here. Okay, now phospholipase C beta was inactive. It wasn't doing anything previously, but now that you've bound this alpha Q GTP subunit to it, that will activate phospholipase C beta, and it's now going to act on a normal component of the phospholipid bilayer, and it's going to break it down into two new molecules. Okay, so let's now discuss this. So what's it going to act on, and what's it going to break it down into? Okay, so it's going to act on a molecule known as phosphatidyl inositol 4,5-bisphosphate, or PIP2 for short. So PIP2 or PIP2 is the abbreviation, but its full name is phosphatidyl. That's the first P in this abbreviation. Okay, and then inositol, that's the I in this abbreviation, and then 4,5-bisphosphate. Now the phosphate, this second P here, this stands for phosphate, and then because you've got two phosphate groups, bisphosphate, you then put the 2 at the bottom. So it's phosphatidyl inositol with 2 phosphates linked onto it. So I want to try and give you some intuition for what this molecule is, and we're not going to draw out the whole formula for it, we're not going to draw out the actual molecular structure of it, but we'll draw out a little cartoon to explain what it is. Okay, so basically it's just a modified phospholipid. So let's start off by drawing a cartoon for a normal phospholipid and then modify it to get what PIP2 is. Okay, so a normal phospholipid looks something like this, as far as my cartoon is concerned anyway. So, we'll start with the main backbone structure, which I'll highlight in green. So this horizontal line that I've now highlighted in green, this represents glycerol. Okay, so this is glycerol. And this is also known, its proper name, its chemist's name, is propane 1,2,3-triol. Okay, and although that's a bit of a mouthful, it's nice because it tells you exactly what it is, whereas glycerol doesn't give you a clue. It's a 3-carbon molecule where you have an alcohol group coming off every single one of those carbons. So the first carbon has an alcohol, the second group has a single alcohol, and the third group has a single alcohol. Okay, and that's what glycerol is. Okay, so then off this glycerol backbone, you then have fatty acid molecules, which are esterified to the first and second alcohol groups of glycerol. Okay, so these are fatty acids that have been esterified. And again, fatty acids have a more proper name. They're also known as long-chain carboxylic acids. That's their more proper name. That's what a chemist would call them. Okay, and again, although this is again a mouthful, it actually does tell you exactly what they are. They're just carboxylic acids which have a really long tail. And basically what you have done is you've esterified these carboxylic acids to the first alcohol, and then you've taken another long chain carboxylic acid and esterified it to the second alcohol of this glycerol. And then you've got these 2 fatty acids which are sort of hanging off the glycerol molecule, and these are known as the hydrophobic tails of the phospholipid. Okay, and then linked to the third alcohol group of the glycerol molecule, so the alcohol group of the third carbon, we then have a phosphate group, which is shown here in pink. So this is a phosphate group, and this is linked via a phosphoester link to the glycerol molecule, okay, to the third alcohol group of the glycerol molecule. And now the entire structure is known as a phospholipid. So the entire thing, which I'll now highlight in blue, is called a phospholipid, and you have 2 layers of these making up the phospholipid bilayer. Okay, and there is an old name for phospholipids, which no one would ever use for just a pure old boring phospholipid, but when we're talking about modified phospholipids, for some reason everyone uses it all the time, and this is the name to call phospholipids phosphatidate molecules. Okay, so another name for a phospholipid is to call it a phosphatidate molecule. And this now gives us some insight into what phosphatidyl might mean here. Okay, so let's now talk about the structure of phosphatidyl inositol 4,5-bisphosphate. Okay, right. So this again is a normal component of the phospholipid bilayer. So not all of the phospholipids in the phospholipid bilayer are just this boring old normal phospholipid. Some of them are modified phospholipids, and we're going to see a lot of these in this video. Okay, so we'll start with this one, PIP2. Okay, so just draw out a normal phosphatidate molecule so far. So here is glycerol again in green. Here are the long-chain carboxylic acids which are esterified to the glycerol in orange. Here is the phosphate group, which is esterified to the third alcohol group of the glycerol molecule in pink. Now, this is a phosphatidate molecule, or a phosphatidyl group, and we're now going to link it to a structure known as inositol. Okay, so inositol is a 6-membered carbon ring, and I will therefore denote it as a hexagon, like so. Okay, which I will colour in blue. Now, inositol might be unfamiliar territory for some people, so I just want to tell you what the structure, what the skeletal formula for inositol is. So remember, in skeletal formulas you don't show carbon atoms, you show them implicitly as corners. Okay, so we've got 6 carbon atoms here making up a ring, and all of the bonds in this ring are single bonds, and then off each carbon what you have is an alcohol group. So you'll have 6 alcohol groups overall, one off each of these carbons in the inositol ring. Okay, and then you'll notice that that only takes the bond number for all of these carbons up to 3. So where's the 4th bond? Well, remember, if you don't show a 4th bond on a skeletal formula, then it's implicitly assumed that that is a hydrogen atom bound to that carbon. So you don't show hydrogen atoms bound to carbons. So if there's a bond missing off a carbon, then you can assume that it's a hydrogen. Okay, so each of these carbons also has a hydrogen atom coming off it. So this is the structure of inositol. Okay, and basically we've taken one of these alcohol groups of the inositol ring and linked it to this phosphate group here of the phosphatidate molecule via a phosphoester link. So the phosphate group has 2 alcohol groups, one that's now linked to the alcohol group of the glycerol molecule, and the other that's linked to the alcohol group of the inositol molecule. Okay, so this creates what is known as phosphatidyl inositol, or PI for short, but we wanted phosphatidyl inositol 4,5-bisphosphate, so we're going to have to stick some phosphate groups onto this structure. So all we need to do is label up the elements of this inositol ring. So we'll label up this carbon up here as carbon 1, and then we just count round in an orderly fashion. So we'll go 2, 3, 4. We want a phosphate group coming off carbon 4, so we'll link it via a phosphoester link to the alcohol group of carbon 4. Okay, so here's another phosphate group. And then 5, we want another phosphate group coming off carbon 5, so we'll link a phosphate group there, again via a phosphoester link to the alcohol group that's coming off carbon 5. Okay, now that is the structure now of phosphatidyl inositol 4,5-bisphosphate, or PIP2 for short. Okay, so what is phospholipase C of the beta type going to do to this poor molecule? And I hope I've convinced you now that this is just a modified phospholipid. It's just a phospholipid with a bigger head, basically. It's got this extra bit stuck onto the phosphate group of the phosphatidate molecule. Okay, but it is just a normal component of the phospholipid bilayer. So phospholipase C of the beta type is going to act on this, and basically it's going to break it down into 2 other molecules. Okay, so it's going to cut. Let me show you where it's going to cut, and I'll show this in turquoise. It's going to cut between this phosphate group and the glycerol molecule. Okay, so the 2 things it's going to produce are a glycerol molecule linked to 2 long-chain carboxylic acids here. So the long-chain carboxylic acids are still in orange, and the glycerol is now still in green, but it's got now a alcohol group of that 3rd carbon that has nothing linked to it anymore. And then the other molecule it's going to produce, so that's one of them, the other one it's going to produce is this inositol ring here, okay, with 3 phosphate groups coming off it. So 1 off carbon 1, 1 off carbon 4, and 1 off carbon 5. So let's colour this in as well. So the inositol ring we'll colour in blue here. Okay, the phosphate groups off the inositol ring we'll colour in pink. Okay, now the glycerol linked to these 2 long-chain carboxylic acids remains within the phospholipid bilayer because the fatty acids, or these hydrophobic tails, are firmly anchored in the phospholipid bilayer. Okay, so this molecule is known as diacyl and then glycerol. Okay, and the reason it's called that is it's a glycerol molecule in green with 2 acyl groups linked off it. Now let me explain to you what an acyl group is. So basically, if you have a carboxylic acid here, okay, so this is a carboxylic acid, then what we're going to do is we're going to link it to an alcohol group on the glycerol molecule via an esterification reaction. Okay, now when you link a carboxylic acid to an alcohol group via an esterification reaction, you take off the alcohol group from the carboxylic acid group, you take off a hydrogen from the alcohol group, and you then link the carbon of the carboxylic acid group to the oxygen of the alcohol group. Okay, so it's always the oxygen from the carboxylic acid that goes off in water, basically. Okay, it's never the alcohol group, sorry, it's never the oxygen from the alcohol that goes off. That's always the oxygen that ends up in the ester link. Okay, so you don't actually add on much of the, well, you do, but you don't actually add on the whole carboxylic acid group. Instead, you just add on this structure here, okay, which I'm now highlighting in turquoise. Okay, now this structure in turquoise, this is what's known as an acyl group. Okay, so that is what you're actually adding on to the glycerol when you esterify these fatty acids onto the first and second alcohol groups. Okay, you're adding on acyl groups. That's why it's called a diacyl glycerol. And another name for diacyl glycerol is to call it a diglyceride molecule. Okay, it's also often abbreviated to DAG for diacyl glycerol. Right. The other molecule you produce is released into the cytoplasm of the cell, and this is what's known as inositol, because it's an inositol ring, and then it's got three phosphate groups off it. So it's 1, 4, because we've got the phosphate off the first carbon, we've got a phosphate group off the fourth, and then finally we've got a phosphate group off the fifth. So it's inositol 1, 4, 5 tris phosphate. Okay, and again, because this is a bit of a mouthful, it's often abbreviated to I for inositol, P for phosphate, and then 3 because you've got 3 phosphate groups. So this is often abbreviated to IP3. Now, in this pathway, we're not actually going to be interested in the diacyl glycerol. We're going to be interested in the production of IP3, because this is going to go and act on IP3 receptors in the membrane of the endoplasmic reticulum, and it's going to cause the release of calcium from the intracellular stores. And we'll continue this discussion in the next video.